Hello, everyone, and welcome to our fifth webinar in the Autism Society San Francisco Bay Area and Parents Helping Parents Spring Webinar Series. We're very excited today to be featuring something kind of new for us. We really haven't done any research-oriented webinars before, so this is the first. We will also have a research-oriented webinar at the end of May featuring researchers from the Mind Institute. So this is very exciting. So today I'm very delighted to be introducing Inside UCSF, latest advances in autism research and how you can help with an introduction to the UCSF STAR Center. Your hosts are me, Jill Escher, President of Autism Society San Francisco Bay Area, and Trudy Grable of Parents Helping Parents. Trudy, say hi.
Good morning, everyone.
Alrighty. Just to let you know, this webinar series has been going on for a little while, and we have several more, as I mentioned. We have 4 of these webinars archived. They're all shown here on the screen. And our next 2 webinars are Social Opportunities for Adults with Autism in the Bay Area. That is going to be a mega panel of 8 different people who provide different programs all over the Bay Area for adults. That should be fantastic. That's May 25th. And then the webinar I just mentioned, May 31st, Inside the Mind Institute. In the fall, we're going to take a break over the summer, but in the fall, we're going to return with an online symposium really devoted specifically to severe behaviors and many, many dimensions. We're talking about treatments, we're talking about housing, we're talking about programs, employment, criminal justice, all sorts of things. So stay tuned in the summer for news about that. Okay, some logistical notes. If you have questions for the panelists, please type them in. You are all on mute, so you can talk all you want, but we can't hear you. So if you have questions, please type them into the question bar, which is in the control panel at the right of your screen. And Trudy and I will pitch those to the panelists, sometimes as they go, but we'll definitely save room at the end. If you're calling in and you can't type in questions, you can email them to me at jill.escher, E-S-C-H-E-R, @gmail.com. Some of you sometimes can't find the presentation screen. If that's you, just look under your browser. Sometimes it's hiding there. As I said, you're all on mute. The webinar is being recorded and will run for about an hour and a half, although give or take. So to introduce today's panel, I just want to mention that UCSF is, of course, a sprawling, large research institution. So when we say inside UCSF, what we really mean is inside just one slice of UCS autism research. UCSF does a ton of other research related to autism that we're not featuring today. So maybe those researchers can join us at another time. But we're very fortunate today to be featuring Dr. Whitney Entz, who directs clinical services at the STAR Center, Dr. Elliott Sherr, who is looking for biomarkers and also doing MRIs to understand brain pathways, Dr. Robert Hendren, who investigates potential therapies and biomedical treatments, and Dr. Ball, who actually spoke at our recent conference in December, some of you may remember her, who is researching one of our favorite topics, which is adults with autism, including the strengths and challenges of adults with limited spoken language. That would be people like my son. So with that, I am going to turn it over to Dr. Entz. Hold on one sec. Oh, wait, hold on. Having a technical problem. Sorry. 1 second, sorry. Okay, Dr. Entz?
Yes.
You're on.
Great. Can you see my screen?
We can.
Okay, fabulous. So I just have about 7 minutes, so I'm going to go as quickly as I can through the different types of services that we have here now at the STAR Center. So the STAR Center means we do service, training, advocacy, and research. And today I'm just speaking to the service the services that we are providing within our center. And we were developed about 2 years ago, really with the goal of developing a multidisciplinary team in order to provide assessment and treatment services for individuals across the lifespan. So to that end, one of our missions is to lead the way to better understand, treat, and promote the well-being for people with autism and other neurodevelopmental disorders. And that's a really important part of our mission, in that we want to serve individuals on the autism spectrum, but then also neurodevelopmental disorders broadly. To meet that goal, we are one of the few centers that provide services across the lifespan. And so we see individuals as young as 12 months, and then adults as well. So we've seen adults up into their 50s and 60s looking for treatment and assessment services. We're an interdisciplinary team with multiple disciplines that work and collaborate together. And our goal is also to be a clinical home for families. So when they come see us, they can always continue to come back to see us either for assessment, treatment services, or just consultation as needs change across the lifespan. So when I say interdisciplinary, what do I mean? I mean that we have a variety of different types of disciplines working with us. We have psychologists, we have behavior analysts, we have social workers, speech pathology, and psychiatry. And within all of that, we coordinate with our research arm, and so we also have clinical and research support in order to collaborate on projects and look at long-term outcomes of our services, as well as just diagnostics and assessment. One of the large things that we do at our center is we provide an interdisciplinary diagnostic assessment. This can be either a first-time diagnosis or a reevaluation, or if families are looking for something for individualized treatment plans and treatment goals. It's a multi-step assessment. So we have the intake appointment, and then we have a number of psychology appointments where the family meets with the psychologist for an in-depth parent interview, and then also direct observation and testing. We also involve speech pathology and psychiatry into our interdisciplinary assessment as needed, and then we provide feedback and also continue to follow up with the families immediately after their feedback appointment to help connect the families to services either within our center or services outside in the community. The assessment components look at activities of daily living, how individuals are doing on their communication, daily living skills, independent living skills. We look at cognitive abilities, and this is measured through a number of standardized assessments, which is dependent upon their age and ability. We also look at direct social communication skills. We look at this through the administration of the ADOS, which is an autism diagnostic observation tool. And we also look at social emotional. So this is especially important as the children become school age or adolescents and young adults, and we really want to tease apart and also be aware of other conditions or symptoms such as depression or anxiety that may be impacting the individual's functioning or just their daily life and daily routines. In terms of our treatment services, our goal broadly is to reduce challenging behavior, and this can be as specific as rigidity or inflexibility. We also help with disruptive behaviors and non-compliance. Another large goal is building social communication and play skills in young children through a naturalistic developmental behavioral approach. Another area we look to improve is increasing peer interactions, and we have a number of different social skills groups to target that. We also heavily focus on improving adaptive skills and how to increase independent living and quality of life. And then also managing emotional and attentional concerns, and this could be through CBT or also medication management services. One thing that we strive to do at UCSF is have evidence-based treatment. So all the treatments we offer, really we strive to continue to use evidence-based practices through our treatments. And we offer a number of different modalities, from individual support, group therapy. We have a heavy emphasis on parent training and parent education. We also offer consultation, and then we have a family support arm in terms of connecting families to additional services and discussing diagnosis and how that impacts quality of life and other services and getting connected. So the models that we use, we use a number of different evidence-based models, ABA, or we also naturalistic developmental behavioral intervention. Under that large umbrella, we specifically have expertise and training in pivotal response treatment, and then the Early Start Denver model. For behavior support services, we use positive behavior support, and we also have structured teaching, and we use behavioral activation and cognitive behavioral therapy, and we are trained in the group model, the social skill group model of peers that was developed at UCLA. So we also, in terms of our group services, I wanted to highlight here that we have a series called the Together series, and this is a series that some parts may fit for families, or they may or one group would just be the most appropriate group. So we have a group for families of recently diagnosed children, and it's called First Step Together, and it's an educational parent support group. And it's a 6-week series where parents increase their knowledge of autism, identify what community supports are, and get some basic understanding of the IEP process and how to advocate or get the needs met in the school and in the community. We also have 2 groups based upon the principles of pivotal response treatment. So we have one group called Motivate to Communicate Together, and then the second group is Motivational Early Learning. The main difference between the groups is Motivate to Communicate really focuses on first word and early word learning, and then the second group builds upon that and elaborates and works on social initiation, so question asking, engaging in reciprocal conversation, really targets motivation, play, and language. It's a group that has about 9 weeks of group didactics and also video-based feedback, where families are bringing in their recordings from what they're doing with the children. But we also incorporate 4 individualized sessions where we're really able to target the individual goals for their child and also give specific feedback. And then the last group is called Let's Get It Together. And this is a positive behavioral parent intervention program. Again, this is a group, but this is split up into half group, half individual sessions where they're learning how to track their child's challenging behaviors, and then we, with the parents, develop positive behavior support plans in order to reduce the challenging behavior and also work on optimistic parenting based on Dr. Duran's work. And it's about a 16-week group. We also have, as I mentioned, a number of parent-assisted social skill groups. So right now we run a number of preschool groups. So these are groups from 4 to 6-year-olds. We have an adolescent group from children aged 12 or 13 to about 17. And then we also have a young adult group, so individuals aged 18 to 35. One of the things that makes these groups different from other social skill groups is that they're parent-assisted, in that the children or adults are in a group at the same time that the parents are in a group. So the parents are essentially the coaches. They're learning the exact same skills as the kids, and this really helps with maintenance and generalization. And it works really nicely with a preschool group where parents are also are coached directly in terms of how to facilitate social facilitation strategies, especially in terms of like play dates. And so then that is something that we really encourage. And based on our needs assessments of the community, we've also found that there wasn't quite a group for children from 8 to 12 years old. So we're piloting a group right now with a parent component specifically for kids 8 to 12 years old. So that's another social skill group that we're offering. And then a really great group that I think that we're wanting to get off the ground very soon is for 14 to 21, and this focuses on the transition age. So how do you transition from high school to college? And that is everything I have to talk about the STAR Center. If you have any questions, there's our website and our phone number.
All right, thank you so much. It's great. I think a lot of people don't know about the incredible breadth and depth of the work you do, so I'm so glad you were able to share that. I am now going to turn the webinar over to Dr. Robert Hendren. All right, this will take just a sec. All right, Dr. Hendren.
Great. Hi, everyone. Should I turn this on to full screen?
Yeah, I think you have to click accept. I don't think we see your screen quite yet.
Got it. Okay. Somehow you're...
There you go. Yep, you're good.
Okay. Well, it's a pleasure to be here. Somehow this thing seems to move on its own, and I'll try and keep it from doing that.
Oh boy, it's like at a disco. Yeah.
Yeah.
Stay screen. Okay.
Okay, good. Thanks. So I'll tell you a little about our Pronto Research Center and then describe one study in a bit more detail to give you an idea of some of the things that we're doing. Our goal is to think of improving the lives of children and adults and families who have ASD, and to do that through better tracking of relevant, meaningful outcomes. This is changing again on its own. This is really distracting. I don't know if there's anything you can do. It doesn't.
There's nothing I can do. It's not based on our system, so it's something weird. Maybe if you take it off full screen. I don't know. Okay. Maybe
or is something sitting on the mouse or the keyboard?
No.
There, that works.
Maybe it'll work now if...
No, it keeps changing on its own.
Do you want to try to... Okay, well, go ahead. See if...
I'll just keep switching back. The things that we do in the Pronto Lab include sponsored trials. We're currently doing a trial sponsored by CureMark that's of a pancreatic digestive enzyme. We have done that trial a couple times, once for kids using a certain parameter and now another. It's a multi-site trial and has been going on across the United States. We're also doing a trial sponsored by Roche. The first one was for adults who had autism using vasopressin with the hopes that it would improve social engagement, and now Roche apparently thought that was positive enough, or the FDA did, that they have now expanded that to be adolescents and potentially children, and also perhaps another adult trial. We're doing a longitudinal study at the Oak Hill School, which is a school for children with autism in San Anselmo that is a non-public school, has right now 36 children who all have autism or autism spectrum disorders. And we measure with an outcome study that parents, teachers fill out about how kids are doing based on case conferences that I go to once a week, that we all work and think about how we can improve the care for children, and to then show in tracking our outcomes whether we're making a difference. We're also using some novel outcome measures that have been developed at the school that are measuring things that they think are the most meaningful outcomes that might be looked at. And then we share the information from what we're gathering with parents, teachers, and clinicians, and hope that in that way, using a kind of medical home model, that will improve our outcomes. The study that I'll tell you more about is a study that we did of sulforaphane at the Oak Hill School. We're also doing research at the Meristem Academy, which is a program for transition age and young adults in just outside Sacramento in Fair Oaks that combines a residential and a community-based program in vocational training for kids on the spectrum based on a model that was developed in the United Kingdom that has been shown to be a benefit, and now we're trying to show that it's a benefit in this beautiful 14-acre campus that they have for kids on the spectrum there. And we're also using the model that we have developed at the UCSF Dyslexia Center, where we're collaborating with the Charles Armstrong School in Belmont to validate phenotypes that have been demonstrated in the lab and then through an outcome study at Armstrong, and are developing an app. So the things that we're doing all relate to using outcome measurements and to building resilience. But I'm going to tell you about one aspect of this in just a minute. Our goal, as I'd mentioned, is to improve resilience and using better tracking of relevant, meaningful outcomes, and doing a better job of partnering and communicating with all the people that are involved using a medical home model. And then we're interested in the development of novel and targeted behavioral biomedical approaches, which I'll tell you about one right now. We have, over the years, been very interested in these resilience enhancing kinds of treatments that might target physiologic abnormalities in autism, things like oxidative stress or inflammatory processes or immune function. Also things like mitochondrial dysfunction, or looking at the health of the gut microbiome and looking at the role of toxins. Over the time that I was at the Mind Institute and here, we've studied omega-3s a couple times, hyperbaric oxygen, methyl B12 a few times, and have published on all of these, and a recent study on vitamin D. These and other things like NAC and probiotics have promise, but are not generally yet targeted for specific abnormalities. People try them for autism, but haven't really said what it is that's benefiting. The early evidence are that there are benefits in changes in these physiologic markers, like in our methyl B12 study, we found that improvement from methyl B12 correlated with increased methionine, with decreased S-adenosyl homocysteine, and improvements in the SAM-SAH ratio. It's not ration, but ratio. But you can see that those were significantly correlated in those kinds of changes, suggesting that these interventions are making a difference in the physiologic processes and how these kids are doing. And this is just getting started as a new way of trying to think about using biomarkers and better targeting treatments in a kind of precision medicine approach. So we became interested in sulforaphane. Sulforaphane is a broccoli sprout extract. It was developed to be used to treat cancer and to perhaps even prevent cancer. It's hugely concentrated. You probably couldn't eat enough broccoli to get what you can get in one of these pills, but it seems to counteract oxidative stress, inflammation, and mitochondrial dysfunction in the previous studies that were done looking at it as a cancer treatment. But it also has been noted to affect heat shock protein. And a person who did the first study, Andy Zimmerman, was one that reported fever being reported with autism. And when someone developed fever in some kids, they seemed to their autism seemed to get better. And so he thought about sulforaphane and heat shock protein and the other effects. And so this group published the first randomized control trial looking at 18 weeks of treatment, showing that in the placebo group there was minimal change, but in the sulforaphane group there was significant change in the aberrant behavior checklist and then the social responsiveness scale. And when they stopped treatment, the benefits went away. You can see a graphic representation of that, looking at the difference between placebo and sulforaphane. On the left, looking at the aberrant behavior checklist, and on the right, looking at the social responsiveness scale. So we thought of studying sulforaphane at Oak Hill School. We couldn't figure out how to do placebo-controlled trials, so we said maybe we could use metabolomics as an outcome measure. Metabolomics kind of come downstream of genomics. If we understand what drives the cell, what drives the physiology, we might start with genomics, but genomics breaks down into transcriptomics and proteomics, and then metabolomics show the metabolic byproducts that are happening as the body's metabolism works or doesn't work as well as it might. And so in our study, we said we'd like to look at sulforaphane and then to see if the benefits of sulforaphane, if we saw any, correlated with those metabolomic changes, that those physiologic changes showed anything that correlated with that benefit. And we hope that this would lead us to a more comprehensive assessment of physiologic changes that could then be used to help us better target treatments. We also wanted to show that we could benefit from an academic school partnership, which we have with the Oak Hill School, and as I'd mentioned, that we also have with the Charles Armstrong School and with Meristem Academy. So we asked all the children at Oak Hill to enroll, and they couldn't have tried sulforaphane or have severe medical problems. We used the aberrant behavior checklist, the social responsiveness scale, and we used metabolomics measured through urine. We were worried that blood metabolomics might have a number of turndowns from kids that said, no, I don't want to do that. But we found all the kids were able to urinate into a little vial that then we could send to the company that looked at the metabolomic changes. And we looked at the clinical changes between those who improved and those who didn't, and what their metabolites were. And we looked also at siblings. 19 patients enrolled, 15 completed. We had 8 people who showed a greater than 4-point improvement in the ABC or in the SRS. And we had 7 who didn't show that change, didn't show a greater than 4-point improvement. So half the people seemed to get better. We also studied siblings, and we're analyzing that now. And we got urinary samples to do metabolomics on the 15 kids that completed the study. Now this is one that you're not going to be able to see, because the problem or the challenge of metabolomics is we got almost 400,000 different metabolomic biomarkers that could be influenced by a number of things that range from diet, but might also have been influenced by the sulforaphane that we gave. You can see all the different metabolomic profiles that we found changed to a significant extent from the group that showed change and the group that didn't. And the pathways that we saw affected involved oxidative stress, amino acid, and the gut microbiome, neurotransmitters, hormones, and a novel pathway that related to sphingomyelin metabolism. We're just now finishing analyzing it. We'll present some of it at MFAR in 2 weeks, but we're getting ready to submit the paper in the next 2 weeks for everybody's consideration. The notion in this kind of metabolic change is that here are all these metabolic biomarkers, but they cluster together in areas around oxidative stress and others that we hope eventually will give us a way that we can determine what metabolic abnormalities might be happening, and could we use supplements in a targeted way to improve the functioning of kids, as we did in the sulforaphane treatment, and show that that improved functioning is affected by the metabolic biomarkers. So that's our study. That's kind of our larger effort. We're thinking now, what are we going to do for our next study at Oak Hill School? We've wondered about the pancreatic digestive enzymes that Curemark's doing. We've wondered about probiotics. We have 3 or 4 other ideas in mind. But we hope that eventually we'll get to the point where we could get a child's metabolic profile and use that to create a cocktail, so to speak, of different agents, different supplements, not drugs, supplements that could help kids function better. We hope will kind of help lift the veil of autism so that we could get a better grasp of the kid that's underneath through the kinds of treatments that we're doing at Oak Hill School. So I think I'm in my time frame, and that's a bit about what we're doing at Pronto.
Thank you.
UCSF Pronto. Thanks.
Thanks, Dr. Hendren. Yeah, well, this is Jill. The most popular quote-unquote supplement these days, at least in my experience in the autism community, is medical cannabis. I'm wondering, has UCSF thought about examining that?
Well, I have a number of kids that I do prescribe medical cannabis for, the kind of CBD, THC combinations. They've gotten more sophisticated. But we haven't yet gotten to the point that we're thinking of actually doing a clinical trial. The cannabinoid pathways would be interesting to follow, but we haven't developed a clear way of doing that. I guess also my impression is that the cannabidiol THC combination is useful for some kids in a modest way in addition to their other medications, and it makes it a complicated study to do. There is a study, that just got started in Israel looking at medical marijuana for autism. Mm-hmm. And there are a few other small open label studies going, and I think actually a major drug company that I'm not free to say right now has developed a CBD THC medication that I think they'll put in clinical trials starting in the fall. So it is of great interest.
Yes, it certainly is. Well, thank you so much.
Thank you for having me.
All right, I am going to turn this over to Dr. Sherr. Let me pull you up here. And yes, everyone, just to let you know, we will definitely be entertaining lots of questions at the end. All right. Sorry. Oh, here you are. Okay. All right, Dr. Sherr. Can you accept?
Can you hear me?
Can
you hear me?
I can hear you. I just don't see you yet. There you go. We see you.
Okay, I just need to find the program to open. Okay.
Yeah, you're good. Hopefully it won't disco out on us. All right.
All right, can you see that okay?
Yeah, fine.
Okay, great. Good morning, everyone. I want to thank Jill and Trudy for the opportunity to tell you all a little bit about what my research group is doing. And hopefully we can get more families interested in what we're trying to do. You can see here, and I'll talk about it a little bit more, our website is very easy to remember, brain.ucsf.edu, and it's reasonably easy to navigate around there if you want to see in more detail what we're up to. So I'm going to talk about just for a little bit some of the advances that have occurred in the last few years, uncovering the genetic causes of autism, and to use that information as a framework for how to think about the majority of kids with autism who don't actually have a single gene cause for their clinical condition. And then I'll just mention briefly again how to participate in our studies. So one of the things that's happened in the last 5-ish years is technology has significantly advanced in terms of DNA sequencing. And all these little figures basically amount to the ability to sequence DNA on a little slide, and you can see that little square with lots of little colored dots, and each color represents a different letter of DNA. And instead of doing one, sorry, there's a background noise. Anyway, so besides being able to sequence this DNA, you can now do this hundreds or even thousands of molecules simultaneously. And so what you see here is the evidence of that from a financial perspective, which is that when we first started doing DNA sequencing, you could sequence a single genome for $100 million. So obviously that was very, very impractical. But like in the case of computers, which was referred to as Moore's Law, there in DNA sequencing, there's been a rapid improvement in the cost of doing this. And so since 2001, whereas it was $100 million to sequence one person's genome, now in 2017, it's $1,000. And that cost is dropping considerably. And that allows us to do things that we weren't able to do before. And so, for example, this is a paper from our collaborators, Evan Eichler up in Seattle. And what this is showing is that if you sequence many thousands of individuals, you see genes that show up repetitively that are statistically significant. So meaning that even though we're sequencing many thousands of individuals, despite that, there are genes that show up again and again. And the ones that are listed in red above the dotted line are examples of that. So I just want to point out one of them, because I'll talk about it a little bit further, is the gene P10 that's up here at the very top. And all of this information has led us to have a reasonably sophisticated overview. We don't know all the details yet, but a sophisticated overview of the genetics of autism. And so that's displayed here. And what the advances gave us in the last 5 years is this blue pie, most significantly, and that's the single gene causes that I just talked about. We also have something called CNVs, or copy number variants, and that's a change in DNA that's not just a single letter of DNA, but usually hundreds or many thousands or even a million or more bases of DNA that's either missing or in certain cases duplicated. So for example, children who have duplication on chromosome 15, something that's called 15q duplication syndrome, it's one of the more common causes of autism, and that is a CNV. And so between the blue pie chart and the red and the green and the purple, which is syndromic, that accounts for about 30% or so. And then we have what we call, that I've labeled here, as polygenic inherited, and that's probably the largest group, and that's individuals where they probably are getting a combination of risk factors from their parents that put them at risk for having ASD. And then finally, there's a smaller group of about 20–25% where we still don't know, and that may represent environmental exposures, gene by environment interactions, and obviously people are working very hard to figure that out. But what I want to do is just take a moment and focus on this 50%, because that's the bulk of children with autism, and if we're going to come up with diagnoses that are medically meaningful and then also come up with therapeutics, it helps us to be able to focus on the largest group of individuals. So again, what I mentioned earlier is that some of this DNA sequencing has pointed us to specific genes, and these specific genes might also be telling us what's going on in the individuals that don't have single gene mutations, but rather have a collection of subtle genetic changes that altogether make them susceptible to having autism. And so what I'm displaying here is a schematic of what we call intracellular signaling. And so what that means is that on the outside of the cell, and the outside of the cell here is on the top of the picture, where these little green diamonds or squares are, and these represent molecules that interact with cells and turn on different molecular pathways. And I've outlined a few of the key molecular pathways that are here. And these are key pathways that tell cells to turn on and off. They tell cells to divide. They tell cells to grow. And they have a lot of sort of fundamental functions in how cells work. And again, I'm going to point out to you just a few of them because I want to talk about them in more detail. So one of them that I've already mentioned is P10, and that's here at the top. There's also AKT, which is another protein that's involved in signaling. And then another one that we're going to look at is down here at the bottom in yellow, ERK, or often referred to as ERK. Now, these molecules are either what are called kinases, or they're called phosphatases. And in the body, cells use these proteins as molecular switches to turn on or off different functions. So if a phosphate group is attached to a protein by a kinase, that's what the kinase does, that's essentially turning on that protein, whereas a phosphatase, as in P10, is able to turn off protein. So again, it's literally like a molecular on and off switch. And what we've hypothesized, and others have as well, is that in a typically developing child, in a healthier state, that there is a homeostatic mean, meaning that there's the right amount of on and off is occurring simultaneously. The cells are healthy, development is healthy. But that if there's either too much turned on or too much turned off of these key signaling pathways, that we see that those children are at risk for having autism. And so just using an example here, the kinase ERK, if it's decreased significantly, we're more likely to have behavioral deficits. If it's increased significantly, we're also more likely to have significant deficits. And in certain cases, you can see this manifested early on in development. So some children have smaller heads, hence microcephaly, and some kids have larger heads, macrocephaly. And both of these are seen at higher ratios in kids with autism. And so what we did is we took a look at some of these key proteins, and this is just a schematic of what some of the raw results look like. And each of these can be visualized on a gel that allows us to analyze many individuals simultaneously. So, and then what this is here is our data looking at the first attempt to study this. And what we did is we studied 12 healthy individuals, and we measured their kinase signaling at 3 different time points. And the idea is that if this is a biologically meaningful measure, that things would not change over time. And so the CV refers to the percentage of variance that occurs in that individual over time in the different measures. And so this is actually a summation of all 12 individuals. And so you can see that the percent of variance is quite small. And that tells us that this is a biologically robust finding, and suggests that we can then look at individuals and see whether this allows us to differentiate between individuals who have autism and those who are typically developing. And so what this is just some of the early preliminary data on this. And this is from approximately 60 ASD children and 60 controls. And you can see here the green is the autism, and the blue is the controls, and this is measuring ERK, and that this is highly statistically significant. The same thing is true for P10, but ended up not being significantly different in AKT. And then we used all of this information to build a model trying to understand how that might allow us to differentiate individuals from who have ASD from typically developing children. And by inputting the full model, we have a value here that's almost equal to 0, and 0 being no errors in our model. And you can see that different pieces of this, whether it be the ERK or P10, that they play a significant role in our ability to differentiate between the two groups. If we have things like gender, we don't see much of a difference, and processing center, which is whether the blood was processed in a core lab or in our own lab, again, we don't see very much that influences that. So this tells us that the model is robust and might help us explain, again, the differences between those who are at risk of having autism and those who are not. And I'll just skip over that. And then another way of looking at it is looking at the sensitivity and specificity. And so area under the curve is just a global measure of the robustness of the model. And so 1 would be perfect, but 0.86 is a highly robust model. Just to give you an idea, some of the main cancer biomarkers that are clinically approved have AUCs that are in the range of 0.6 to 0.65, so much, much lower in sensitivity and specificity than our preliminary model appears to be. Now, other folks have been thinking about this as well, and we were pleased to see that a paper came out at the very beginning of this year from folks in Ohio, Craig Erickson's group, where they looked not at all of the different kinases that we looked at, but just ERK, and they also found a difference between neurotypical children and children with ASD. And so we interpret this with enthusiasm to suggest that somebody has at least partially replicated our findings, suggesting that this is a significant way to move forward. So summary and future directions. So ASD is biologically and phenotypically heterogeneous, and that creates sort of a need for greater understanding of the underlying biology. And despite this heterogeneity, it looks like at least some of the individuals show differences in kinase signaling, even though they may not necessarily have genetic differences in those particular genes. The ones that we focused on today were RAS MAP kinase, or otherwise known as ERK, and the P10 pathways. And there may be others out there, and we need to look at that further. This is stable within individuals. We've only tested this in controls. We'd like to test it in children who have autism as well to confirm our initial suspicions. And again, I mentioned the prior work and the impact that has, I think, on validating this approach. So one of the things that I didn't really talk about, mostly for sake of time, is the age of the group of children that we tested. We tested a broad range of children from sort of ages 1 through preteen. But what we'd really like to do is test children who are pre-symptomatic. So whether that's children that have early signs of autism and the clinicians are not certain, or whether their family members already have autism and there's a slightly increased chance that they would develop autism. And so our ultimate goal is to use this kind of tool as a way of early identification of somebody who might be at risk, so that either behavioral treatments or supplements or other medicines that become available that we can use these in a judicious way in children who would benefit the most from that kind of intervention. And I've only talked to you today about a few proteins, but we're collaborating with a lab here at UCSF that can use a tool called mass spectrometry, and that would allow us to measure the abundance of, instead of 3 or 4 proteins, we can do dozens or even hundreds, and I hope that we'll have some interesting preliminary results to report on shortly. I haven't talked about it today, but we're also building a research program to use brain imaging as another way to identify children who are at risk, and to potentially use that brain imaging as a concordant biomarker for improvement. So it's sometimes difficult to measure clinical improvement in children. It takes a long time to really be confident that that's happening, but it may be the case that certain imaging metrics would allow us to see those things sooner, and then therefore evaluate the effectiveness of approaches more quickly. And then finally, we're seeking participants. You know, one of the things that I want to make sure we do whenever we engage in any sort of trial is to, if at all possible, provide benefit back to the families. And so since we're doing clinical testing, we can provide information about that back to the participants. And we also are setting ourselves up so that we can do genetic testing on all the participants, as I know that's something that's of interest to many families. So again, our website is brain.ucsf.edu, or you can contact me directly or my study coordinator, Talia Burson, whose information is listed below. And I just want to say that the work that I've been talking about today is done by the 4 individuals listed here, who are really fantastic. And it also is worth mentioning a heartfelt thanks to all the families who've participated in our studies to date. Thanks very much, and I'll be happy to take questions when we get to the Q&A.
Okay, thank you very much. Just quickly, you mentioned that you were moving towards genomic testing. Is that this year, next year, who knows?
Yeah, so we're collaborating with a program that's called SPARK, which is through the Simons Foundation, and they plan to do DNA sequencing of 50,000 families with autism. And so we are, we hope to be up and running in the fall of this year for that program.
Got it. Okay. Trudy, let's take a little tiny question break. I know I'm showing my screen now. I can't find the screen. Oh, dear. So let's take a little question break while I look for my screen. And actually, no, I'll queue up Dr. Hoosball while we take some questions. Do you want to go ahead, Trudy?
Yes.
Okay.
So there are some questions. There's a question from Sarah asking, what type of brain imaging can shed light? Are there studies for teen, 17 and over in age?
So the short answer is yes, we are enrolling teenagers, and there's a number of different tools that we're using that both address the structure of the brain and as well as measuring how the brain, for example, processes language. One of the things that we've shown is that in groups of individuals with autism, that the rate at which sound, particularly sound in the form of language, goes from your ear to the part of the brain called the auditory cortex that processes that information is actually slowed, and that might be an important reason why children with autism can have language difficulties.
Thank you.
Okay. Another question for all of the panelists. For adults, and actually we hear stuff like this a lot. For adults with autism now in their 30s who also have epilepsy, what advances in evaluation and testing, i.e. labs and imaging, in metabolic, genetic, and neurological domains might be of benefit in optimizing their health care? And I should just say, a lot of us who have older kids, for example, genetic testing has advanced a lot since they were first diagnosed, let's say, 15 years ago, in my case, more than 15 years ago. Where should these people go for, getting more advanced testing, genomics, and also the things that the questioner asked?
So I can go ahead and start on this, and others can chime in. So just to by way of further introduction, I'm a child neurologist, and so I take care of children with autism who also happen to have epilepsy in a lot of cases. I think that there is a subgroup of individuals who might have specific genetic perturbations that both make them at risk for having autism and epilepsy, and understanding the reasons why might allow you to select a particular kind of medicine that could optimize your child's outcome. And so if you're already seeing a neurologist for that, then I would just ask them to help you do that by something called whole exome sequencing that's clinically available. A number of commercial labs offer that. And just as another example, I spoke in my talk about a group of kids who have duplication on chromosome 15, 15q dupe. It turns out that those individuals have what we would call basically that they have subtle but clinically not perceptible seizures. So they're electrically seizing, but their body's not moving while they're sleeping. And that treating those children with an epilepsy drug, even though they're not having physical seizures, can again both allow them to sleep better, have better better opportunities for learning because their brains are not being disrupted while they're sleeping, and this often can have both behavioral benefits as well as, again, better acquisition of language.
All right, Trudy, do you want to ask one more question, and then we'll move on? And the questions can be of any of the panelists.
Yes. Is it beneficial in an individual healthcare context to do genetic testing for an adult with autism? And does UCSF do so on an individual basis? That's for anyone.
Yeah, does anyone else want to take that? Otherwise, I'm happy to.
Go ahead.
Okay. So I think that's a great question. I think that some people would argue that the utility of this test is, still to be proven. Just a priori, if the person has autism, you're likely to get an answer doing genetic testing about 20 to 25% of the time. So we think that that's a significant, advance. We think that even if there's not an immediate, change in medication treatment, that there may be lots of other opportunities to better understand the condition by having that genetic information. It allows you to reach out to other families who have similarly affected kids. And quite honestly, a lot of times the most important insights in these conditions are coming from moms and dads who've raised their children and are astute observers, and they can share this information with each other much more readily when you have a specific genetic diagnosis.
Okay. Let's say you were a parent and you just didn't know who to call. Who do they call? And you wanted to get this done.
So are we talking about an under 18?
Oh, a genetic test.
Yeah, so at UCSF, there is an exome clinic that is run by my colleagues in genetics. And I can, after the webinar is done, look up the exact website address to direct people to so that they can contact the clinical coordinator if they want to make an appointment.
Okay. Yeah, we could post any links on our website, on the webinar website as well. I have a couple other questions that came in, but we will save them for after Dr. Hoosball's presentation. Okay, take it away.
Hi, everyone. Thanks so much. I want to start off and just thank Trudy and Jill both for organizing and inviting me to be a part of this webinar today. So I'm going to talk to you about my lab, which we've called the Lifespan Lab. And let's see. Okay, and so the Lifespan stands for Lifespan Symptom Profiles, Achievements, and Needs. And the focus of our research is really to advance understanding of adults with autism, and particularly to think about informing clinical practices and supports. So to do this, we have research that spans kind of three broad areas. The first of which is trying to understand autism symptom profiles across the lifespan. So in different developmental periods ranging from pretty early development all the way through older adulthood. The second area is looking at factors that may influence outcomes. So most often our work is looking at factors, individual factors such as cognitive or language abilities, and also starting to broaden out to think about pubertal development, emotional processing and functioning, and then also collaborations to think about how the broader sort of context, so environment around them, may be affecting development and longer term outcomes. And then the third area is really focused on adults and trying to understand the various range of achievements and needs that adults with autism are both achieving and facing as they are going not just into transitioning into young adulthood, but also through middle and older adulthood. And for those of you who follow autism research, you probably know there's very little research in this area, and so that's something that we're really looking to expand. And so across these three areas, we have several projects that I'll give you a little highlight in the next slide, but really looking to improve the methods that we use both in our clinics and our research projects to assess what we call clinical phenotypes, or basically the behaviors or symptoms that individuals find are both supporting a diagnosis and maybe interfering with daily life. We try to integrate a range of different approaches, looking at both biological indices as well as parent report, self-report, and direct observations of behaviors. And we're really trying to advance understanding across the range of cognitive and language abilities. So you'll find a couple of the projects that I'll talk about are particularly focused on individuals who have limited spoken language or intellectual disability. So this is just an overview of some of the projects that we have going on currently in the lab, some of our data that's already been collected and we're trying to analyze and publish papers, and some are projects that are actively recruiting. And I'll talk about a few of these in more detail in a minute, but again, trying to just kind of give you some context for some of the research that you may hear about. There's a large international meeting for autism research that's happening here in San Francisco in a couple of weeks, and so hopefully there will be some press about some of the new research that's coming out, not just from UCSF, but around the world in autism. And so just to kind of give you some context for what we're doing in our little corner of the world. So again, in terms of autism symptom profiles, when I'm talking about improving autism assessments, looking at the measures that we're commonly using in clinics, as well as trying to develop new measures, both to improve our ability to diagnose autism, but also to attend more to some of the strengths. I think there's been a lot of focus on understanding impairments and challenges, and it really helps us if we also have a strong sense for individual strengths to be able to build treatment programs and better support people. and thinking about characterizing autism across the lifespan. So what does autism sort of, quote, look like in toddlers versus school-age children versus adolescents and adults, and looking at how autism symptoms change across different developmental periods. And I'll show you a little bit of data from an ongoing project in a minute. Thinking about factors that influence outcomes. Again, really trying to understand across the range of abilities, but we have a few projects that are focused specifically on characterizing minimally verbal children with autism and understanding different cognitive profiles and emotional and mental health. And then also several collaborations looking at different biological mechanisms. So specifically, looking at the relationship between specific genotypes, like Dr. Sherr was discussing, and the, what we call the phenotypic outcome, or the behavioral manifestation of symptoms, and how that differs across different genetic disorders. And then also looking at physiological emotion reactivity, which is looking at things like cardiac and respiratory and skin temperature and electrodermal activity in response to emotions, and how that might be a mechanism or an underlying factor of the symptoms that we see in our clinics. And then finally, in terms of the adult achievements and needs end, two projects. One is particularly looking at strengths and challenges of adults who have limited spoken language, and we're really trying to focus on expanding definitions of outcomes. So this is what we would call a pilot study, and we're trying to gather information from families that have adults with limited language to move us beyond focusing specifically on whether individuals are living completely independently and in a post-secondary or a full-time job, and having significant others or other kinds of relationships, which is how the research world has traditionally defined outcome. And as a result, many of the adults with limited spoken language have been put in sort of a category of not having very good outcomes. But that doesn't really help us in terms of how we should move forward in terms of supporting adults, as we're trying to understand what is a meaningful outcome across the range of ability levels. And then the last project there in the bottom right is just looking more broadly. We're setting up an online study now to look at neurodevelopment and neurodiversity across adult life. So starting with young adults all the way through older adulthood for those individuals who are both diagnosed or suspected of being diagnosed, and trying to understand various areas of stability and change throughout their adult lives. So before I go on to talk specifically about the projects, I just wanted to kind of highlight, I have a wonderful team here at UCSF, and so this is work that I have the good fortune of being able to lead, but is strongly supported by the three young women on the bottom of the screen, and also in collaboration with a number of individuals, both across different departments at UCSF and around the world. So I wanted to just present really quickly two studies, one of which is being presented actually at the International Meeting for Autism Research in a couple of weeks. So this is sort of hot off the press. And I think it's important to just kind of throw a little bit of data out there so that when families are thinking about whether they want to come into the lab and participate in the studies, you have sort of a sense for, what are we doing with all of the hours of questionnaires and assessments that we're asking you to do. So on this slide, we have the three graphs. Each line is a different question about nonverbal social communication. So each line, one line might be gestures, one line might be facial expressions, and all different symptoms from a parent interview used to assess autism. And so higher scores indicate more definite presence of the symptoms. And these questions were asked of 132 children who were diagnosed with autism at age 2 and followed until they were age 18. And so each of these graphs shows changes in these symptoms for children grouped by their language level. So on the left we have, so all of the children were language delayed at 2. On the left, the verbal group, those are children who had phrase speech at 3 and were verbally fluent by age 18, so you can think of them mostly as sort of age-appropriate levels of spoken language. The delayed verbal group in the middle is children who were speaking in single words or less at age 3, but by 18 either had developed phrase speech or maybe speaking in sentences. And then the group on the far right, the delayed minimally verbal group, had single words or less at age 3 and continued to be minimally verbal by age 18. And so what I'm showing you here is really that across these different language groups, we see different what we call symptom trajectories. So if you look at the verbal group, many of those symptoms are going down, or so the presence of those symptoms or the severity of those symptoms is decreasing over time. But if you look closely, there's a couple of lines that are relatively stable across the age range, and even one line in red that's sort of showing a U shape. And you can see that across each of these different graphs, each of the different groups of individuals who show different sort of patterns of language development, that those trajectories of the various symptoms are differing across time. And so, the takeaway point from here is just that autism symptoms are changing as children get older. So some are relatively stable across childhood and adolescence, others are improving with age and probably cognitive development, and the level of improvement may vary by language level. And I put improvement in parentheses because I want to really emphasize that the majority of these children, all except for 7, continue to meet autism criteria at age 18. So even though we're seeing improvements in individual symptoms, based on an interview that is really looking at symptoms that are related to diagnosis in children, what this tells us is that we maybe don't have a clear picture of what autism symptoms look like in adults, or in some adults, particularly those who develop more language. And so we really need more research to identify what are the symptoms that characterize autism in adulthood to make sure that when we have new measures, so that we can develop new measures that are capturing symptoms in adults, particularly those adults who are coming in for a first-time diagnosis in adulthood, and we may not have the luxury of having a really comprehensive developmental history. And I think, for clinicians who might be listening, it's really important that we're all very cautious when we have an adult who's presenting for a first-time diagnosis, that we don't assume they don't have autism because a classic autism symptom, like an abnormality in gestures, isn't present. Because it may be just that we, if there was an abnormality present earlier in life, by the time they've come to our clinic, that's something that has improved or developed over time. So the second study I wanted to highlight was published last year in the fall. And this research was focused on characterizing minimally verbal children. And so there are really 3 main findings I think are sort of takeaways from this study. The first was that how you define minimally verbal matters. And so the 5 different colors across your screen right now are just different instruments that we use in autism research. And so the specifics of the instruments don't matter so much. But what I want to highlight is that depending on what instrument you use to define minimally verbal children, you end up with different subgroups of children. So if you are using the instrument on the left, the ADOS, which is the Diagnostic Observation Schedule, a direct assessment, and you're using the language observed during a 45-minute period of the ADOS, to quantify minimally verbal children, to classify minimally verbal children, then you're going to get a larger group, and you're also going to get more variability in that group, which makes sense because we know that when we bring children to clinics, it's a new situation, and we may not hear all of the language that they're capable of using. On the other hand, if you use the instrument on the far right, which is the Vineland Adaptive Behavior Scale, to ask, and this is a parent interview when we ask questions about language level, and use that to classify children as being minimally verbal, you get a much smaller group, and you also get a much more homogeneous group, in that these are the most impaired by parent report. And so these groups all differ both in terms of age, as well as their level of language and their cognitive abilities. The second point is that even within the group defined as minimally verbal on an instrument. So again, here's from the ADOS. We can further divide into different subgroups based on even how many words they're using in a single session. And that, so on the left, we have children who are using 5 or more words in this 45-minute period. And on the right, you have children who are using, it should be 0 to 4 words. And so, I think for many of us, if you think about whether a child used 1 or 2 words versus 6 or 7 words in 45 minutes, it might not seem like a meaningful difference, but this division actually does seem meaningful when we actually look more closely at the characteristics of the children in that group. And so just really highlighting that there is a lot of variability even within the broader group of children that we would call minimally verbal. So those children who are using a few more words tend to have somewhat higher cognitive abilities, somewhat better language skills by parent report, as well as somewhat higher adaptive behaviors, so daily living skills and social skills. And then the third finding from the study, and I think which was most important to highlight, is that there might actually be different reasons that children remain minimally verbal. So on the right, you have children whose verbal skills are roughly equal or commensurate with their nonverbal problem-solving skills. And we think this even profile suggests that for those children who are minimally verbal, language development was probably affected by their intellectual disability. So their verbal and their nonverbal IQs are about equal. On the other hand, if we look in the left, in the column in green highlights a group that shows a significant discrepancy in verbal and nonverbal problem-solving skills. So in other words, their language seemed to be much more impaired than what we would expect given their estimated nonverbal cognitive level. And so somewhat surprisingly, these groups didn't differ significantly on any of these other measures that we looked at. So autism symptom severity, other measures of language, or adaptive behaviors. And so this led us to think that there was something else that may be interfering specifically with the language development of children with this specific cognitive profile that wasn't actually their intellectual capacity. And so this study, just to kind of summarize, how a study defines minimally verbal is going to affect their results. And so, the different definitions are going to include different subsets of children. And so if we really want to understand the broad range or variability that we see in children who we might refer to as minimally verbal, we really need to be careful about our definitions and how we're selecting our samples. There's a lot of variability within that minimally verbal group that really remains to be explored and understood. And I would say, at least based on data from this one study, about half of the children seem to have much higher nonverbal problem-solving skills than expected, given their level of spoken language. And what that suggests is, clinically we need to be very careful about assessing abilities to better understand strengths and not assume impairments in other areas are equal to their language level, but also that we need to continue to do longitudinal research to better understand what interferes with language development in this subset of children if it's true that their language is not meeting the level of cognitive capacity that they seem to be showing. So, with that in mind, hopefully getting a taste for some of the research findings might excite some families to start to get involved. So I can tell you just about a couple of studies that we're, one of which is actively recruiting. So this is the Exploring Strengths and Challenges of Adults with Limited Spoken Language that I mentioned. So we're looking for individuals who are over the age of 18 and have challenges with spoken language. So it might range from being completely nonverbal or using single words up to using simple phrases to communicate. Some individuals who've come in certainly are quite proficient with using augmentative communication devices, and that's great. We're really focused on the level of spoken language in terms of defining recruitment. And so we have people come in for an in-person visit, and we do a series of activities with the adults and some interviews with the parents if they're willing. And families get a $50 gift card just to try to compensate for travel time and costs to get here, and a brief summary of some of the results. So if you're interested, you can contact the lab at the email on the screen or the phone number there. And like I said, this study, so the one aim of the study is to try to understand symptom profiles in minimally verbal adults, and we're developing a new measure to assess autism symptoms in older individuals. But a big focus of the study is really on expanding definitions of what is a good outcome. And so, you know, thinking about, again, kind of reaching past some of the traditional adult milestones that are used as sort of where we've set the bar, but thinking about sort of you know, more individualized outcomes. So how good or how strong is the person-environment fit? Are these individuals in environments that, you know, are good fit for them? You know, what about level of independence, both in terms of being able to do things for themselves around the house and in the community, as well as to be able to advocate for themselves. Physical health, I think, is a very important one, as we know that influences many other domains of your life. And then thinking about more subjective things like happiness and emotional well-being, and both how can we measure those things and how can we you know, sort of better assess them so that we can develop supports and programs to be able to promote positive well-being. And if you're not quite ready to participate in a research study, or you don't have a family member that would qualify, you might know, again, I mentioned the IMFAR meeting is going to be here the second week of May, and UCSF is hosting a one-day meeting prior to that conference that's going to focus on measuring meaningful outcomes. We have a full-day program, including 4 keynote speakers that will talk about a range of different outcomes in school-age children and adults, as well as 2 panels comprised of researchers, educators, clinicians, individuals with ASD, family members, and other stakeholders. And they're all going to provide perspectives on what are meaningful outcomes and hopefully generate discussion with all of the participants who come. And so the day is really set up to be kind of like a brainstorming activity, again, to get us to push the boundaries of what we think of as a meaningful outcome, which we hope will really inform research and clinical practice. So if you're interested in that, you can visit the STAR Center's website for more information about registration. And then just one quick last plug. So we have one upcoming study that's looking at the role of emotion processing in autism, and we'll probably start recruiting sometime in the early summer. This is focused on totally opposite age range, so looking at 7 to 12-year-olds with autism. And you can see my little cartoon there. We're looking at physiological correlates of emotion. So what that means is the child would come up to UCSF and sit in a small lab and watch some videos on the computer screen. And they would get hooked up to different sensors that monitor heart and breathing and skin temperature and those sorts of things. And so we're focused primarily on children who are verbal and who can speak, you know, phrases or sentences, and who have the ability to sit in the lab and watch some videos for about an hour and a half to 2 hours. So that's all I have to share. Again, thanks so much for listening. And if you have questions or comments or have interest in any of the research studies, our website's on here, as well as our contact information. Thanks so much.
Thank you, Dr. Husball. Appreciate it. I know, yeah, go ahead. Go ahead.
We've had a lot of questions about supplements based on these biomarker studies. Looking at these issues, are there anything that you're seeing that help with signaling issues, metabolic pathway challenges, or others? And that's for anyone.
Trudy, I'm not sure I understand the question. Oops, hello?
Let me try that again. So there are several questions about supplements for people with autism. because of this robust study marker, what types of supplements are they looking at?
You're asking for Dr. Sherr. You're asking.
Thank you. Hopefully he hasn't had to leave yet. But there are several other questions related to supplements as well.
So to answer the question as it's related to the work that I showed, we're not in any position to make those connections. We know that there are certain molecules and certain conditions where these pathways are affected, but there's not any evidence yet that particular supplements favorably move these pathways in children.
Okay.
Thank you.
I'm wondering, Dr. Sherr, if somebody enrolled their kid in your study, might you have information that may become relevant for intervention parameters later? I mean,
that's the optimistic cross your fingers hope. But I don't want to falsely raise expectations. That's our goal, but we're not there yet.
This is a question that we don't usually like, kind of personal clinical questions, but I guess this would be for you, Dr. Sherr. The child has a CHD2 variant, but no visible seizures. With medication for epilepsy appropriate?
So sorry, what was the question?
The child has CHD2 variant. I don't know what that is.
That's one of the genes that I talked about.
Oh, okay. And would no visible seizures, but would medication for epilepsy be appropriate? Asks this mother.
Gotcha. I'm not aware that CHD2 has that connection that I spoke about, for 15q, for which there is a lot of evidence. One thing that I would say is, that's something to discuss with your neurologist. It's not unreasonable to think about doing an overnight EEG if it's not too traumatic for the child, to see what it looks like.
All right. I have a question. Dr. Hendren, are you still with us?
I am, except that I can't, I'm not sure that anybody can hear me.
You're pretty dim. We can kind of hear you, I guess, if you scream, or I don't know what happened to your headset. The question is about sulforaphane, and where could people obtain sulforaphane, and whether it's safe to try without a physician's supervision.
You can get sulforaphane through Nutramax is the name of the company. It's a supplement, and people can take it without a physician's prescription. They talk about how to dose based on weight, and the thing that Nutramax sells is the one that we used in our study. There are a few other companies that make sulforaphane as well. I think Kirkman Lab does, I think a few others, but Nutramax is the one we used in our study.
Okay, thank you.
That work?
All right. Yeah, we kind of heard you. It was pretty good. Thank you. Trudy, do you want to try one? Uh-oh, I think we seem to have some audio problems. I'm going to go ahead and ask another question. I'm a neuropsychologist who assesses children and young adults for autism. Traditionally, interventions recommended have been focused on psychobehavior, milieu, and educational interventions. But what I'm hearing is that it is increasingly helpful to recommend child-adult neurologist genetic testings and consideration of supplements, meds that address the individual's particular genetic profile. Can behavioral pediatricians play this role as well? I guess, you know, the main gist with this question is what we hear all the time is like, where do I go to figure out what's best for my kid? You know, who are the clinicians that can really provide guidance? No answer?
So I think this is Elliott Sherr. The short answer, I think, would be that it's a team approach. And I think that as we learn more about autism, the team that's going to help is going to expand.
Okay.
and not one clinician can typically can provide all this expertise and guidance that's necessary.
But the point of entry for a family could be anybody on the team. It could be the neurologist, it could be the geneticist, it could be the behavioral pediatrician.
Yeah, all of them can be the point of entry, and the best clinicians are going to be the ones that bring a whole team together to answer the question.
Okay.
I do think it's a challenge for families to figure out where is the place of entry, and I think if their question is for genetics, which was kind of what this question had to do with, then I think entering with a geneticist or with a neurologist like Dr. Sherr, or there's a few others that specialize in the genetics part of autism, is a place. Psychiatry does that, but not in the same frequency that might be done by a geneticist or by a neurologist that is specialized in that area. There are certain treatments that one might find more in one place or another, and I think it's really hard for families to navigate their way through. We've tried as much as possible here at UCSF to have a good collaborative relationship, so we refer patients back and forth and we say this is a better one for you, and I think that helps when you go to a medical center and have the opportunity for people to collaborate.
Yes. Okay, let's try to get to a few other questions. Trudy?
Yes. How do you recommend for the layman to differentiate between the groundbreaking research that UCSF is doing versus the endless noise of pseudoscience ASD families are subjected to? For example, cannabis has potential positive effects on ASD. I have seen naturopaths recommend CBT as well.
Is the last one CBD?
CBD, yes.
Yeah, okay. I think it's, I try to sit down with families and look at the evidence, look at the literature, and not by Googling the term. I suggest often that they can go to pubmed.gov and put the terms in that they're interested in and see what published studies have been peer-reviewed and are reasonable to consider, what's the evidence. And so I think trying to help families become a better educated consumer, I think can often help people figure that out. But if there's no published studies and no good review of the mechanism that's been published, then I think it's less likely that it's going to be something that they ought to be eagerly pursuing.
All right. Very quick question here. Somebody asked about getting a discount as a parent to the MFAR conference that will be in San Francisco. My belief is no, there are only discounts available to students, and not to, there's no parent discount. It really is intended as a research conference. It's not really intended to be a family conference. Nevertheless, you can go on the website and call them and ask. You never know if they have some special kind of scholarship program. I just don't know of one.
And our Wednesday pre-conference is $25.
Which is a lot cheaper because MFAR is like $500 or $600.
Yeah, the $25 is less than what the coffee costs us for people that are there. Yeah.
Right.
And also the pre-conference, sorry, this is Whitney speaking, is meant for families and community members to attend, so that the Wednesday day is probably the best day for community members to hear a little bit more about just measuring outcome and looking at that as well. And it's quite a bit cheaper than the MFAR conference.
Yes, definitely, and probably easier to understand at several points as well. All right.
And we thank the Autism Society, for the Bay Area Autism Society, for their support, and for others who have been supportive. So thanks.
Yeah. It's not every day that MFAR comes to our backyard, you know. Yeah, like almost never. So it's pretty exciting. Okay, here's another question, and then Trudy will wrap it up with one more, I think. What is the prognosis? It's impossible to do a prognosis during a webinar, but what is the prognosis for ASD children with high cognitive and language intact but social pragmatic communication and peer engagement challenges, no gross motor or fine motor issues. I don't know, Dr. Husball, Dr. Entz?
Sure. So I think at this point, to be really honest, we don't know, and that individual outcomes vary really widely. Certainly, we know that language, having functional language by the age of 5 years, and having average or above average cognitive abilities are important strengths, and those individuals maybe have better odds of doing better, but I think that the level of the social, communicative, and then pragmatic challenges varies so widely within individuals, and it's a very hard thing to predict. And also, for some children do quite well because they have a number of environmental supports. Early on, and it's not until they're transitioning to adulthood or adolescence or adulthood when other things start to crop up that also interfere with being able to achieve the kinds of goals that they're hoping to. And so I think even on an individual basis, when we see children clinically, I think we're very cautious about making longer term predictions, and that even in children with typical development who are going to a pediatrician, you don't know kind of what other things can also sort of crop up across the lifespan and interfere with the kinds of plans and hopes and dreams. Certainly, again, I think the most positive outcomes that we've seen are for individuals who have language and cognitive strengths, but there's a lot of variability.
Okay. Trudy, do you want to ask a final question?
Jill, I need to sign off. I'm going to let you manage.
Okay. All right. Well, thank you. All right. I will pitch a last question. There's a question here about training, and if UCSF provides training for the direct service providers who work with the adults with autism. So one problem that we have here in the trenches is we have adults often with very difficult and challenging behaviors and intensive needs, but care providers are often very poorly equipped and also very, very poorly paid to work with them. So this mom is asking if UCSF fills any of that need.
That's a great question. I think at this point we have a number of training programs for professionals, but I'm not sure it's quite what the caller is asking about. What I would say is, for the person who's asking, please feel free to reach out to the general STAR email, or you can email even my lab email, and I think we do have a number of collaborations across the Bay Area, and we're very interested in trying to support professionals. We may not be able to do it right now, but I think hearing about what the needs are in the community, we're very dedicated to being a new program, to continuing to develop our services and programs to meet the community's needs. And so it's helpful for us to know what gaps are out there. And it may be, depending on individual needs, there may be something that we can do to help support.
Okay, and there's just one more question. I'm going to squeeze it in here because it's so related to that other question. This is from another mom. This is also, I guess, mostly for Dr. Entz and Dr. Husball. Have you found that your assessments at UCSF have helped enhance regional center services and supports for adults with autism?
So I can start, this is Whitney, I can start speaking to this question. Vanessa, you're more than welcome to jump in. What I have found is that we do conduct the assessments for individuals over 18, and many of those individuals have been turned away from regional center previously and they weren't allowed to, regional center wouldn't do an assessment because they were already over the age of 18.
I think in this case, just to squeeze in this case, this is for somebody with obvious autism and was diagnosed as a child, but is in need of greater services and support as an adult.
We have found that Regional Center is accepting our reports, and they do use them to help with services, but every regional center is different. So we've just had experience with Golden Gate and East Bay, but that they are accepting of our reports and recommendations, but of course they are restricted in terms of some services that they can offer, but we do our best.
Yeah, the only thing I would add to that is I think we have a great team, and particularly our social worker who really helps support families who come through for evaluations, even after the evaluation, and, tying in with various service agencies, regional center and otherwise, and she's very knowledgeable sometimes even about the kinds of language that needs to be used to help advocate for some of those services that are needed, and so I think in that case, the evaluations, maybe even somewhat have better chances of being well received by different providers, because I think, knowing kind of what services are available and the ways in which to advocate for them.
Yeah, it's really interesting just from a parent's perspective how many evaluations you get for your kid, your autistic kid, and then you base your advocacy based on those, reports, professional reports. Then when they become an adult, you don't really do that anymore. But they're still even in greater need, right, of supports. And I think we don't do enough to evaluate our kids, reevaluate them as adults. A lot of people have written in to ask if these slides, if the show will be available, the webinar will be available online. Yes, it is being recorded, and it will be sent to everybody who registered. You will get a link, and it will be posted on our website. Okay, so I am going to wrap it up. I want to thank our panelists. We really kind of covered a dizzying array of topics, from biomedical interventions to biomarkers and genetics, to evaluations and clinical work and adults with autism. I mean, it kind of ran the whole gamut, so sorry if your head is spinning, but I was really pleased to be able to present these wonderful researchers. So thank you for what you're doing, and I am going to call it a day. Thank you, everyone.